Yes. Yeah.
Perfect. Well, if I disappear or there are any problems, do just let me know. Otherwise, I'll just get started. Great. Thanks, Max. Great. Well, thanks very much for coming. And I volunteered to talk about multi-scale models of the human brain. So I'm going to talk a little bit about how we construct models of the human brain and how we use that to simulate, particularly in this case, ischemic stroke, but also talking a little bit about some other things too. So I guess I don't need to persuade everybody of the importance of cerebral blood flow, not particularly this group of people. So obviously we care about the rising clinical importance of cerebral disease, particularly the long-term care implications of those. And particularly now people are much more aware of the role of cerebral blood flow in dementia and how that's part of the cascade that leads towards Alzheimer's disease. And at the bottom here, I just put some simple statistics looking at the death rates in England and Wales, men on the left, women on the right. And you can see how the big rise in dementia and Alzheimer's is a big societal problem. And so trying to understand how cerebral blood flow goes through the brain and how it's monitored and how it's measured and how it's controlled are very important. And although I'm not going to talk a huge amount about control, I did want just to make the point that the brain is one of the most tightly regulated organs in the body, which is very important in terms of how it behaves. 12 to 15% of the body's cardiac output and 20% of the oxygen consumption. And even though its metabolic requirements remain largely static, it's very tightly regulated in response to, say, arterial CO2, neural activity, arterial blood pressure, and sympathetic activation. So there's a lot of things that are acting to control blood flow in the brain. And so you can see the importance of blood flow in cerebrovascular disease. So particularly the importance of localized behavior, particularly when we think about stroke and dementia, and the importance of the link between the structure of the cerebrovasculature and its function. And alongside that, there are a lot of blood vessels in the brain, and we can only image down to vessels of about 1 millimeter in diameter. So we can only really see the large blood vessels. And so trying to image all the blood vessels in the brain and trying to model them all is really an impossible task, or likely to remain an impossible task for the time being. And so, as well as the fact the system is active and dynamic, means that this is a really complicated mathematical problem. And what I'm going to do today is talk a little bit about how we go about approaching this and how we go about modeling cerebral blood flow in humans. So what I'm going to do is talk a little bit about the physiology and how we image it, just to sort of set the background, because that's quite important in terms of how we do the mathematics and set up all the models, because we want to make sure the models actually are based accurately on physiology. And I'll then spend most of the time talking about flow and perfusion models, but I will talk a little bit about how this can be extended to oxygen transport, to water transport, and to cell and tissue death models, because these are all obviously a key part of the INSIST consortium and what we're planning to do, but also the wider picture in terms of modeling the response to ischemia and to stroke. And if I have some time at the end, I'll finish off by talking about a little bit more about cerebral blood flow control. And on the right here, I have some images from this classic 1981 paper, which is really the first time that people had started to look in detail at the cerebral circulation. And these are taken from post-mortem human brains. And these were injected and then imaged. And so a lot of interesting data came out of these brains, and I'll return to these a bit later. But I just put this up to highlight the complexity of the cerebral circulation. And of course, as I've just said, it's a highly complicated system, and it also bridges a wide range of length scales, and that's really important in terms of what's coming up in a moment. So on the left, you can see this is a paper from 2013 where they imaged the large vessels, so 6 colors for the 6 territories, middle, posterior, and anterior cerebral arteries on the left side and right side. And then you can see these large vessels then move to the pial circulation, which you can see on the middle slide. And then on the right-hand side, you can see that these then turn into descending arteries, ascending veins with a capillary bed in between. And even just from this, you can appreciate the huge complexity and the huge number of blood vessels involved in perfusing the brain. And of course, imaging is a key part of understanding what's going on. And here, put a couple of pictures of MR angiography looking at the flow in large vessels. And of course, that's really important, looking at the flow in the large vessels, because that's where we tend to get clots. So that's where ischemic stroke is really critical. And we can look at the flow in these individual large vessels, but you can clearly see from these images that you only get down to a certain diameter of vessel beyond which you really just can't get any kind of resolution at all. And there's about 1 millimeter before you reach that point. And on the right, you can see some perfusion imaging done using CT in this case, and can also be done using MRI, looking at perfusion imaging. Often you'll use a contrast agent for that, although modern methods, something like ASL, actually means that you don't need to inject a contrast agent, but you will get a very low signal to noise ratio. And so again, you can see you get information, but you're not looking at individual vessels. You're getting some measure of what's being perfused through individual volumes of tissue. And so you have this fundamental limitation about what the resolution is that you can get on your imaging. And that provides quite a constraint on what we can measure, but it also provides a constraint on how we model and what information we know about the cerebral circulation. So the multi-scale modeling is based upon this idea that we can separate out what we call length scales. So on the left-hand side, you can see a sort of a macro scale picture of a brain, and that's rather like the MRA pictures I had on the last slide. And you can see that at that large scale, you can image a certain number of vessels. You can see the circle of Willis in the middle and then all the cerebral arteries coming out. But then if you go along the top, if you zoom in, then you can actually start to see a little bit of the detail, which will be the vessels that you can't quite resolve, but are quite close to that resolution. And then if you keep going to the right, you can then see that you zoom in again. And then finally, as you get onto the right-hand side and you zoom into the finest level, you can then actually see the capillary scale, and you get the smallest vessels. And then along the bottom, you go back up through the venous circulation. So the idea here is that you can do this separation of scales, and you can go all the way from the capillary bed on the right to the large vessels, the large arteries on the left, and in between you've got this separation of scales. And that's very important mathematically. What I'm going to do is just use an example of where you might see that somewhere else. So here's a road map of the UK and Ireland. And at this scale, you can see the very large roads. So you can see the large motorways that go around the UK. And you can see the ones that go around Ireland and going to our good friends in Galway on the far left-hand side. And of course, at that length scale, I can get a certain amount of resolution, but there's an awful lot I can't see. I can't see the road in which I live, for example, on this map. And then if I zoom in, so I come in and zoom in on the Google Maps, and now I can start to see the road system around London and Oxford, and I can see a little bit more detail. So on this map, I would be able to work out how to get roughly to Oxford. And if I continue to zoom in, I can now start to see finer detail. And so at this length scale, I can now start to see this more local roads around Oxford. And once I zoom into this length scale, I can now start to see most of the big roads within Oxford. And then finally, if I zoom in to the absolute finest detail, I can now see all the local roads around the bit of Oxford in which I happen to live. So you can see at this scale, this is a bit like the capillary bed. I can now see each individual vessel, and it's hugely complicated, particularly in Oxford with the one-way system. But as you zoom out again, you can then start to appreciate how these roads then become like A roads and motorways rather than like the large arteries, the arterioles. And so if you're trying to navigate your way around the UK, if I was trying to drive from here to Birmingham, I wouldn't care about every local road. I'd only care about the large roads getting from here to Birmingham. And then once I got to Birmingham, I'd care about the details of how to get to the particular address. So this gives you an idea of what I mean by multiple scales. It's like zooming in and zooming out on what I'm looking at. So in the circulation, I'm zooming in from the large vessels to the small vessels, just like in here, I'm zooming in from the large roads to the small roads. So that's the sort of the guiding idea behind how we approach this. But mathematically, what how do we actually go about doing this? And we use a technique called homogenisation. And this was developed to model water flow in soil, essentially. So it's been around quite a long time, and it's used in a whole variety of different problems. And it's based upon the idea that a lot of materials behave differently at different length scales. So if you zoom in to a very small length scale, a material will behave differently to the way it will behave if you zoom out to a large length scale. So for example, a laminate material or a porous medium, they will behave very differently at different length scales. And I'll show an example of that on the next slide. So what we really want to do is to describe a composite material at a global scale, removing the local variations. In other words, how do we describe how this behaves at a large scale, which is on the whole what we really care about, and not having to worry too much about local variations and local fluctuations at what we would call a local scale. And this means that the governing equations will behave differently at different scales. And what homogenisation does is allow us to couple the two together in a mathematically rigorous way that enables us to solve at the global scale, in other words, sort of at the whole brain level, but include all the detail about the local scale of each individual capillary. And solving, as I put here, solving these flow fields would be very difficult to solve. And in fact, in the brain, it would just be impossible to try and solve. If I constructed a network of every vessel in the brain and tried to solve the flow field through it, it would just be impossible. So what I'm going to do is use a couple of examples to illustrate what I mean by homogenisation and how we apply it. So if you look at the figure bottom right, so the guy is holding this block of material, and you can see that water goes in at the top and it comes out at the bottom. And so it's not completely solid. So this would be known as a porous medium. In other words, water can flow through it, or a liquid can flow through it from one side to the other. And what we care about is if I put a certain amount of liquid in one side, how much comes out the other, or if I put a driving pressure across it, how much flow goes through it. And a sort of schematic of that is on the top. You can imagine here's my rock with gaps in it, and I put a high pressure on the left-hand side and a low pressure on the right-hand side. And what I care about is, well, how much flow goes through it. And you can see someone's gone to a great deal of effort to put in all these individual bits of the rock and then to solve the flow field going all the way through. And it looks very pretty, and it takes a huge amount of computer time. And actually, you could probably just say, well, all I really care about is the relationship between the pressure on the left and the pressure on the right and how much flow goes through it. And that's a very simple relationship. I don't need to know about all the details of all the little rocks in between, and I don't greatly care most of the time about all the little details of how the flow is going through. What I really want to do is to summarize how that piece of rock is behaving. And again, on the bottom left, you can see an example where someone solved the three-dimensional flow field through a piece of rock. And this is interesting and potentially quite useful, but it means that it's immensely complicated trying to understand the flow patterns, and the detail we don't really care about a lot of the time. If I'm interested in a stroke, I don't greatly care about every single capillary vessel in the brain. So what we want to do is to characterize the behavior, and that's what I'm going to come back to in a moment. So we want to characterize the behavior of this rock, or in our context, the behavior of the brain. And so this kind of approach with homogenization is used in flow through soils and rocks. It's also very commonly used in fracking and carbon dioxide sequestration, which are two big topics of the day. So fracking, so if you look at the top, is when you basically try and extract oil from the ground by pumping in lots of high pressure water and driving it out. It's very controversial, but it's very well funded. And there's a lot of interest in how you develop models of this to try and extract the most amount of oil for the least effort. So the oil and gas industry funds a lot of work in porous medium and homogenization methods because they're interested in how to extract oil from all these tiny little cracks that you can see on the right. And as before, we don't greatly care about each individual detail of each individual crack because we can't see them from the surface. What we care about is the overall properties, and that's what we care about in the brain as well. And on the bottom, you can see, sorry, you can see how CO2 is injected into rock because it's being stored there, and you can see how it passes through the rock. And again, we want to understand how CO2 passes through the rock and how it's therefore stored. So there's a lot of interest in these kind of models from the oil and gas industry and also from the sustainability industry. So a lot of the techniques I'm going to talk to you about actually equally well apply to this, and there's a lot of crossover for between the different fields. So at this point, I'm going to do a quick mathematical aside. This is going to be quite brief, but for those of you who are interested, I just want to go through a little bit about how homogenisation works and how these multiple scales methods work. If you are interested in this, then the book on the top right is the one I would really recommend, by a guy called Holmes, Introduction to Perturbation Methods. This is what I recommend to my undergraduates when I give this lecture course. So I talked a little while ago about the separation of length scales and this idea that one length scale is much bigger than another. And that's how I get from the large blood vessels down to the capillary bed. And this naturally lends itself to being a perturbation method approach because the perturbation methods are based upon this idea of a small parameter. So what I'm going to do is do a couple of examples just to illustrate how this works. And I picked a really, really simple equation. This is just a cubic in X, and I want to solve for X. And of course, I could do this numerically, but what I want to do now is to illustrate how I can use a perturbation approach to do this. So I said I need a small parameter. So what I do is redefine 0.001 as my epsilon. And epsilon is what I always use for a small parameter. And you'll see that again when I put some of the results up from what we've been doing. So we have this parameter epsilon. So I've now rewritten my equation in terms of epsilon. Now, the key thing about a perturbation method is that because I can assume epsilon is small, I then assume what's called a perturbation series, that I assume my solution is a function of this epsilon, this parameter in my equation. There are no other parameters, so it's just this epsilon. So I assume it's a power series. And this is a good approach because epsilon is small, so these terms become very small very quickly. So once I've done that, I can substitute it in. Oh, sorry, I have a tickle in my throat. So I can substitute it in, and I get this rather unpleasant equation. That's always one of the nasty things about perturbation methods. You always end up with one horrid equation. So once I've done that, I have this equation. You think, well, how on earth do I go about solving this? It looks horrid. So what I do is I say, well, my solution, this equation is always zero. It must solve whatever the value of epsilon, because I've made this equation a function of epsilon, but it must solve whatever the value of epsilon, which means that all the coefficients of these terms must be zero. So I can, this coefficient is zero, this coefficient is zero, this coefficient is zero, and so on and so forth, however accurate I want to be. And that then gives me enough equations to solve, and I can end up with the solution x1, x2, x3, and I get 3 solutions because it's a cubic equation. So it's a very, very simple way of approaching a problem. You set up this parameter epsilon, which is a small parameter, which is what I already talked about. Stick it in the equation, collect all the terms, and out pops the answer. The good thing is actually that packages like Mathematica, so any kind of symbolic package will do this automatically for you. So in fact, there are shortcuts. You don't have to do all of this by hand. So this key idea, the first key idea is that we have this small parameter in our solution or in our governing equation, which we then use to formulate our solution. We can extend this to differential equations. And the key concept here is that we can now use multiple scales. And this really harks back to what I was talking about before. So I can say, here's my differential equation. This is just a very simple second order differential equation, and I want to solve it. And if I solve it numerically, or in fact, I can solve this one analytically, it's very simple. With given boundary conditions, I can find the solution, which is the solid line on this plot. The approximate solution is the one we're going to find in a moment. So the exact solution, and you can see that it changes quite quickly in one part of X, which is along the bottom, and quite slowly in another. And that's a characteristic behavior of multiple scales. So what I do is I say I've got two scales, X1, which is X over epsilon, and because epsilon is small, this basically amplifies X into X1. So this zooms in to the bit where it's changing rapidly, and X2 looks at the rest where it's slowly changing. And then just like I did before, I assume a power series in epsilon. So I assume Y is made up of a series of terms in ascending powers of epsilon. Stick it in, do a lot of processing. This will take me about 10 minutes, and out drops the answer. And the answer is a very good approximation to the original solution. And in fact, we can put as many terms in this as we wish, and we'll get more and more accurate answer. This is just a pretty good, a simple approximation to this, the solution to this differential equation. But you can see in the solution, it's dependent upon both X and X over epsilon. And that's a key property of these equations based upon multiple scales. So they're based upon X, which we could think of as the large scale or the global scale, and X over epsilon, which is like the local scale where we zoom in for the very small behavior and all the small changes. So then we can extend this to what I called homogenisation. And the key concept here is that we take our local scale or the small scale and we average it out. So if I take sort of very simple second order equation, so this is the diffusion equation. So D is my diffusion coefficient, just like I might solve for oxygen transport in the brain. And I can say this second order differential equation. And where D, so the diffusion coefficient, is dependent both upon the global scale or the large scale X, so this would be like the whole brain scale, and X over epsilon, so zooming in to my local scale. Because in other words, the diffusion will depend upon precisely how all the capillaries are orientated and how they line themselves up. But that behavior, I don't really want in my big equation. So what I need to do is basically average it out. So just like before, I do a power series in epsilon. And this one will take me about 20 minutes. I'm going to skip through. And what we end up finding is that we end up with this average diffusion coefficient, and this D with the bar over the top basically has averaged out all the local behavior so that I can now write the equation down, and U naught means I'm only solving the first term in U, and therefore I can solve this equation based upon D bar, as we call it, which incorporates all the information from the local scale. And mathematically, we can calculate D bar basically from the local scale, but you don't need to worry too much about the details. That just comes out from working out how the diffusion coefficient varies locally. And as long as I know how it behaves locally, I can do some averaging. And essentially, this is an averaging that enables me then to solve the equation on the global scale, but include all the information on the local scale. So all the little local variations and bits and pieces that happen at the capillary level get averaged out. And so essentially homogenization allows us to average out all of this behavior in a mathematically rigorous way. And we'll see how that works as we progress. And this is a paper that Wabi and I wrote when he was a graduate student, looking at how we homogenize blood flow in the cerebral microvasculature. And I just put this up to illustrate that basically all we're doing is exactly what I did on the previous slide. We have our network, and we can see because at the local scale, which is the little d, the small little square, you get a certain type of behavior. But what we really want is the behavior at the large scale once we start putting these all together. So in other words, as we start to zoom out, how does this behave at the global scale? And you can characterize this in the equation basically like a porous medium, it turns out, and that's what Wabi proved in his paper. that we can actually homogenize this out, and we end up with what's called this permeability tensor, which is this K in the equation. And that relates the flow to the pressure gradient. And we can calculate the permeability tensor, as I'll show you in a moment, based on the capillary vessels and their behavior. So that's the mathematical aside. So I'm now going to start to think about how we actually do this for the human brain. So this is the slide I showed a few minutes ago, and this is just to remind us that this is what we're doing. We're separating out the length scales. On the left side, we have the macro scale, and on the right side, we have the capillary scale. And then we have these various levels of scaling all the different blood vessels up from left to right, and then on the arterial side and on the venous side as well. So the separation of scales and the approach that I showed on the previous slide means that we can actually write down the equations for each one of these and build our models all the way from the bottom up. And we work our way from the bottom up because we now know a lot of information about the capillary vessels. And this comes back to that classic paper I showed you from 1981, right at the beginning. It was 25 years later, the same group basically took all of those casts and basically quantified. They used a lot of very smart imaging and analysis tools to characterize the behavior and summarize all the properties of the microvascular network. And you can see how complicated that is just from those two figures on the left. So this is all experimental data from human casts. And what they did was convert this into properties like the frequency distribution of diameters and the frequency distribution of lengths. And they published this in a paper in Microcirculation in 2006. And when I read this paper, I realized that we could use this information to construct generic capillary cubes, and we could do that in arbitrarily large number and therefore characterize how the cerebral circulation behaved. And after a lot of work and Robbie's hard work on his DPhil, we finally managed to get this to work, and we published this 2015. And these cubes are now quite famous within my research group. Everybody uses them. So we generate these capillary beds, and we end up with these capillary structures. And you can see the tiny length scales is about 400 micrometer cube. And these cubes match all the experimental data that's available on them. So they match all the experimental properties, all the connectivity, all the diameter distributions, all the length distributions of the data that was published in 2006. And there have been some follow-up papers since. So we can construct this, and therefore we can then calculate the permeability, just like I talked about a couple of slides ago. And so using these cubes, we can actually calculate the permeability. And this means that we've moved up, we can move up one length scale. And so on the left-hand side, now notice this is millimeters. So this is now a cube. This is a voxel size. So 2.5 millimeters by 1 millimeter by 1 millimeter. And so we've generated all the descending arterioles and the ascending venules in blue and red. And the pink is now the capillary bed, but the capillary bed we're not modeling as individual vessels, we're modeling it as what we call a continuum, because it's modeled by that permeability tensor that we calculated on the previous slide. So the pink stuff is all the capillary bed, and that simulates the flow through the capillary bed based upon those capillary models. So it's like at every point in this space, we have one of those little cubes I showed you on the previous slide. And this means that we can actually now solve the flow field at a voxel scale, because previously we would not, if we had to put every capillary vessel in here, there would be tens of thousands and it would just take forever. Trying to solve the flow field through very large connected networks is very, very tiresome and very computationally expensive. So suddenly you can now solve the flow through these networks at this length scale much more easily. And just as an illustration, we then blocked one of the penetrating arterioles, and you can run simulations, and this is looking at the drop in pressure as a result. And you can see how we end up getting this pyramid or inverted pyramid of pressure drop, which matches up very nicely with animal experiments, although we haven't done any explicit validation. And I would say that all our models are based upon human data, whereas the one on the right is actually animal data. So our networks explicitly encode all the properties of human cerebrovascular networks. So having done that once, we can sort of do that again. We can just keep working up, doing multiple separations of scales. And basically at every scale, you solve the flow equations explicitly in the large vessels and use a porous medium for the smaller scale. And you can just keep averaging up and working your way up towards the surface, and you end up with a whole series of equations, which, as you can see, all look pretty similar. And so we can solve for the flow field all the way up now to the large vessels. And this makes it mathematically rigorous, but also computationally tractable, because it's actually doable now, rather than just trying to solve everything. So once we've done that, I'm going to start to think about how we actually implement this. So here's our brain. This is a slice through a brain, and you can see the grey matter and the white matter, and then the ventricles in the middle. And we have an arteriolar compartment, and a capillary compartment, and a venous compartment. And these exist at every point in space. So every point in space has some kind of averaged arteriolar compartment, some kind of averaged capillary compartment, and some kind of averaged venous compartment. And then we can couple the equations together by conservation of flow. So flow goes around each compartment, and K is the permeability of each compartment. And then beta governs how easily the flow goes from one compartment to the next. And you can see it goes from arterial to capillary, and then from capillary to venous, as you'd expect. And so we then need to start to think about these parameter values, because we've talked a little bit about permeability. So now we need to think about the coupling between the different compartments, because of course that's not something we've solved for previously. And so this is what Tamas did when he first arrived. And this is very neat parameter optimization work that he's done, trying to work out how do we calculate these parameter values. So there's a lot on this slide, sorry. On the left side, that's the arteriola. So these are the descending arterioles. In the middle, we've got our capillary bed. Those are the famous cubes. And on the right, we've got the ascending venules. So you can see how we've got, and every point in space, of course, is an averaged or characterized version of these. Now, the arterioles essentially act to push flow down, so they have a permeability in the downwards direction, and we assume it's governed purely by that, and everything else is just random distribution. The capillary, we know, is in every direction. It's what we call isotropic, which means that it flows equally in each direction. So it's a diagonal matrix up on the top right. And then the venous circulation acts to move the flow back up again, so it can be drained and go back through the sinuses. So, and we calculate Kc because we know how the capillaries behave, because we've got Wabi's cubes and we can characterize them, which means that we then end up with 6 parameters in our model. We need the permeability of the arterioles, the permeability of the venules, and then these coupling coefficients, and we assume that they're different in white matter and grey matter, because the flow field's fundamentally different in those, and that's an important thing to consider in the model. So we then make a few assumptions, because that's quite a lot of parameters to optimize over. We don't really like 6, that's too many. So what we assume is that the coupling coefficients scale between grey matter and white matter, it's the top left. We then assume that the venous permeability is twice the arterial permeability based upon experimental data. And we get a first approximation of the arteriolar permeability from this tube bundles approach, which gives us a rough idea, and then gives us a rough basis for what the ratio is between arteriolar and capillary permeability. And that gets us down to 4 parameters, which is a much more tractable optimization problem. So these assumptions make our lives a lot easier. And there are some constraints upon this, because we know roughly what pressure drops are, we know roughly what flows are, and we know that everything must be conserved in the steady state. And so this basically enables us to match the perfusions in the grey matter and the white matter, and to ensure that perfusion is conserved. And then we can use some pressure distributions to get an idea. This is experimental data, so which we've used. So this gives us the pressure drop from left to right as we go along the circulation. So we go into the capillary bed and then out through the venous circulation as we go from left to right. So we can then use this data, get a rough idea of the pressure drop, and then we can get an estimate of the coupling coefficients in the grey matter. And so that then enables us to get down to just two parameters that we want to optimize over. So the ratio of the coupling coefficients in the grey matter and the white matter, and the ratio of the permeabilities. And then we can optimize this with a particular cost function, solve it numerically, and this is, you can see we get a very nice convergence in both the grey matter and the white matter, and they converge on the perfusion values that we are expecting, which is great. And the cost function drops to a very low number, which means that we're converged. And we can then see that it converges. So on the top one, you've got the ratio of permeabilities, and on the bottom one, you've got the ratio of the coupling coefficients, and you can see that these converge very nicely. And we'll see what the results look like in a second. So that means we've got the model, we've got our parameter values. So the next thing we need to do is to think about how it applies to a real brain. And I'm just going to go through this relatively briefly, but I want to make the point that actually implementing this is not trivial, and actually working out how to implement this in a full brain model is not a straightforward task, because there's a lot of things you have to consider. And this is really some great work that Tamas has been working on the last couple of years. So perfusion. So on the left, you can see that these are the penetrating vessels as they come into the brain. And you can see that they follow a general direction in towards the middle. And what we've done is set up a nice field approach that enables us to map the descending vessels into the brain, coming perpendicular from the surface and then moving in towards the ventricles. And we can then take, using MRI data, we can take a mesh generation of the brain. And this is a seriously non-trivial task, trying to mesh everything, trying to filter out the bits of the brain you don't want, set up the right boundaries, make sure everything's continuous and everything's nice and well behaved. And extracting the brain from the skull, converting it into grey matter and white matter, so that you can see the grey matter and the white matter, using a package called FSL, which is really good for this kind of thing. And that then enables us, and this is a huge amount of work, which I'm skipping over really quickly, to generate our whole brain model with grey matter and white matter along the top. And you can see the three standard ways that we slice through the brain. And you can convert that into a finite elements model on the bottom left. And this is what we then use in our Phoenix solver. So we then have our model, and I've shown some of these results before, and this is work that we've been doing with Raymond and Alphonse, and they've been doing some very nice work on modeling the flow in the large vessels, which is why I haven't really talked about that at all. But you can couple the model together, because we can now add the large vessels to our model, and that gets us to the surface of the brain, and looking at perfusion territories, which enables us to map out where the different blood vessels supply different regions of the brain. You can see the red is the anterior circulation, the blue is the middle cerebral artery circulation, and the lighter blue is the posterior. And you can see all the different territories mapped out, because that's important, because you need to know where the flow from the large vessels actually reaches the brain. And again, I'm going to skip over this quite quickly, because partly because probably some of you have seen it before, and partly because Raymond and Alphonse have really taken the lead on this great work. So, and they'll be presenting another time. So I don't want to present everything they're going to present. So but you can see the perfusion territories and the permeability. And you can see on the left this directional map that I talked about. And then that enables us to map the permeability at each point in space of the brain. And that then enables us to actually solve the full flow field based upon our homogenized multi-scale model. So really that just highlights how variable the permeability is in different regions of the brain, depending on which direction the flow is being pushed. So then the numerical implementation, so you can see this is a sort of healthy brain, this is a perfusion map, and you can see that the perfusion is very uniform in the grey matter and very uniform in the white matter. They're very different, of course, but they're very uniform in these two volumes. And so this is a very normal and nicely well-behaved behavior, but you can see how the model gives you a nice, well-behaved perfusion in both grey matter and white matter, thanks to the way the model is constructed. And of course, then if we're interested in ischemic stroke, so a right middle cerebral artery occlusion then introduces a large perfusion change, and you can see the perfusion deficits in purple, and you get very large perfusion deficits. This is quite an early simulation, and there's no collateral flow in this, which is something that Raymond is working on at the moment, and the results obviously look very different depending on the level of collateral flow, but this is just a very early result to highlight how we can solve the perfusion flow field in the whole brain model. And looking at the clinical comparison, you can see the CT images, you can see the dark sections, and you can see on the top left, you can see this large region, again on the top right, and again on the bottom left. And actually, if I go back, you can see that those correspond really well with the predictions here. And so this would be for a patient with very, very poor collateral flow. You can see actually, even just looking at them, the results really are in pretty good agreement. So the agreement for perfusion is actually really quite good. Now this is I think this is, I can't remember if it's an anterior or posterior cerebral artery occlusion, but this is a smaller vessel occlusion, and you can see again, you get a very different style of perfusion deficit. Again, purple is the perfusion deficits that you see. So you can see how you end up by simulating the perfusion and simulating the flow in the whole brain. You can see you now start to examine where the deficits are. And something that's really interesting that other people are working on at the moment is looking at how collateral flow affects that, and then thinking about how do we validate the model at different levels of collateral flow, and how do we relate, in fact, the results to the collateral flow. And again, this is just a sort of a slightly different way of showing it. On the left, you can see the healthy brain, and on the right, you can see the middle cerebral artery occlusion. It's worth highlighting that the perfusion in this is actually it does vary even in the healthy scenario, which is what you would expect. So there are variations in what you see in perfusion and flow in different regions of the brain. So that wraps up the flow section. What I want to do now is think a little bit about oxygen transport and how that can be solved in a very similar way, because all the mathematical tricks that we've used for flow can be applied to the transport of oxygen through the brain, which of course is critical because it's hypoxia that leads to ischemia in brain tissue. So I've taken you all the way from the capillary bed up to the large vessels and back again, and showed how you generate these models of perfusion and flow in the whole brain. What I'm now going to do is think about oxygen transport, and again do this at multiple scales. And I've drawn the diagram slightly differently here. At the top, I've got the large circulation coming down to the pile circulation, then the descending arterioles, the capillary bed, and back up on the venous side. The reason I've put this in here, again, because we now have to consider it's not just a capillary bed, because it's not just a matter of flow. We now have to consider the fact that we've got neurons, astrocytes that need to be fed with oxygen. And that's, of course, the whole point of bringing oxygen around the brain, that we want to get it from the capillary bed to all the brain tissue. But I also put this up to highlight that the solid arrows which govern flow of blood also basically drive the flow of oxygen. And so the oxygen model is heavily coupled to the flow model. And also there's water transport, which I'll talk about quite briefly a little bit later. So you can see we've got our model for blood flow, but we've also got oxygen flowing around the system. And that's a coupled system. And as we did for the flow, we can again use the multiple scales approach. We can zoom in and zoom out, looking at going from the large scale on the left-hand side to the capillary scale on the right-hand side. The equations for oxygen are much more complicated because it's a coupled system. So in fact, I've only put two of them down here, and you can see they're much longer. There's nothing much harder to solve mathematically. It's just that they're more complicated equations. But it's a very similar approach, which is why I won't go through that in any detail. But again, we need properties of these equations. And so you can see some of these things that are underlined or have bars over the top. These are average properties. So these mean that we're having to use our small scale models to calculate these values. So for example, D2 in the third line is something that we need to calculate from the capillary networks and then scale it up. So This is something that one of my graduate students is working on, finishing her thesis at the moment, and calculating all of these parameters. So for example, surface area to volume ratio and the diffusion coefficient, how that's averaged over the capillary bed. So again, we need to do parameter estimation and generate simulations based upon the same anatomical model. So the advantage is once we've got the flow field, we can then calculate the oxygenation. And you can see some preliminary results on the bottom right. So this is the oxygen in the capillary bed calculated for the whole brain model based upon the flow model that I showed you a moment ago. I also showed you how we were doing the optimization for the parameter values for the flow model. And we're still working on that for the oxygen model because there are some parameters that we need to optimize over, just like Tamas did for the flow model. And this is ongoing, so I don't have many results to show you from this, but this is a very similar approach mathematically to solving for the flow field. To solve for the oxygenation field, we can do basically exactly the same thing. It's just the equations are a little more complicated. And it turns out the optimization it also is a little bit more complicated because the model turns out to be non-linear, which means that some of the tricks we used before are a little bit more complicated to use. So that's one of those things. Also, having talked about that very briefly, I'm just going to mention a little bit about water transport, because we obviously care about the transport of water, and because edema is very important in ischemic stroke. And at the moment, our model doesn't really consider this at all, which means that if we're going to look at stroke and do validation, we can't consider any patients where there's significant edema. So whereas that is actually very important in a lot of patients, and particularly telling the difference between cytotoxic edema and vasogenic edema, as you can see in the bottom. So these are different things, one where the cells swell, so the intracellular space increases, and one where the extracellular space increases. And these occur after stroke, and these can occur in different proportions in different regions of the brain, and it's very important to understand how these behave. And they're of course driven by hypoxia and driven by ischemia. So this is something that we're working on at the moment, and I have another graduate student looking at multi-compartment models of oxygen transport coupled to multi-compartment models of blood flow, and then coupling that to edema and looking at water moving across the blood-brain barrier and leading to swelling either in the intracellular space or the extracellular space. But again, it's all based upon these very simple models at a very small scale. So here's just a single capillary vessel. And so if we can understand how water transports itself across the blood-brain barrier at this length scale, then we have the mathematical tools to scale this up. And I have another graduate student who's working on these mathematical tricks, so we can actually scale up using homogenisation and scale up from the capillary bed all the way up to the whole brain model, and therefore generate a set of equations that we can couple with our blood flow model and our oxygen transport model. At the moment, this is pretty much a multi-compartment model rather than a multiple scale model. So it's a much more simplified version, and we're working on developing the multiple scales version. This is based upon the work of a guy called Penta, who's done a lot of work on the mechanical properties of brain tissue at multiple scales and done some really nice studies on characterizing how brain tissue moves at a global scale, depending on the properties at the local scale. And that's something that we're also interested in exploring in the future. So this is work that's very much underway, although one of my previous graduate students did do some very preliminary simulations where we simulated a stroke, and in that ischemic region, we assumed breakdown of the blood-brain barrier. And that enabled us to predict a certain amount of water leakage into the tissue. And we got a certain amount of cell swelling. And we ran this on a very simple brain geometry and looked at how the brain tissue moved, and actually got pretty good agreement with the experimental data that we found for brain tissue. This is something we'll be coupling into the full brain model that we developed and I've described a minute ago, although it's not part of the INSIST program. So this is something we're doing on the side. And then of course, finally, what we really care about is whether the cells and the tissue actually survive this ischemic stroke, and whether they survive and how much of it survives long enough to either survive or to recover after either thrombolysis or thrombectomy. And I put this slide up quite regularly to remind myself how complicated the process is that leads from ischemia. So if you look in the bottom left-hand side, you can see I've got blood pressure driving perfusion, and perfusion drives oxygen and glucose supply. And that then drives tissue oxygenation, tissue glucose, which then provides ATP, which then drives neurons, and that then enables the vasculature to remain in a healthy state. But if there's anything wrong with that, so if you get a clot, then the permeability, so this is the green bit, so it says blockage. If you get a change in permeability, that will affect your perfusion. Then if your oxygen drops, you can get pericyte constriction, which means that the small vessels, the capillary vessels, will start to constrict because pericytes die in a constricted state, and that affects capillary permeability. And then you can get inflammation, edema, and endothelial dysfunction and vascular smooth muscle damage. So all of these things can interact to predict how your tissue is going to damage, and some of these things can be reversed and some of these are irreversible. So we start very simply by looking at the effects of vessel occlusion. And on the top left, I've got a healthy brain. This is just a slice. I should emphasize these are all 3D simulations. It's just for ease of plotting. Often we just plot a 2D slice to make it easier to look at. So again, we've got our perfusion, and you can see the perfusion. This looks very similar to what we had before, but with different colors. So the perfusion in the gray matter and the white matter, you can see is quite different. And on the top right, so we occlude the RMCA 100%, and you can see we get this huge, great deficit in perfusion, as you'd expect. And that's similar to the results I showed you a moment ago. Obviously, that isn't in reality very likely, and this is assuming there's no collateral flow, but this is a very sort of simple baseline for comparison. On the bottom left, what we've then done is occlude penetrating arterioles, and we've occluded 10% of them. And the occluding at this level doesn't seem to make much difference. Whereas if we occlude on the bottom right 10% of the precapillary arterioles, in other words, if we go slightly further downstream, then you get quite a large deficit. And you can see there's quite a large deficit in perfusion, which corresponds to the RMCA occlusion. Obviously, it's a smaller deficit, but if left unchecked, then there's the possibility for that to turn into a large perfusion deficit, and then eventually to turn into infarcted tissue. So where the occlusion occurs is really important, unsurprisingly. So understanding how that behaves, particularly in the smaller vessels, is very important, because precisely where the blockages occur makes a huge difference to the actual result and the actual outcome. So we spent a lot of time, or Wabi did, spent a lot of time looking at the effects of blockages on the microcirculation and on how this affects hypoxic and ischemic tissue. And so for that, we've gone back to our characteristic cubes. So what we've been on the top left, you can see, again, this is another representation of our very small cubes. Again, remind you, this is all based upon human data, and these are converted into these synthetic networks that match all the experimental properties. And what we've then done is simulated oxygen transport at this level, because we're interested in what happens at this length scale in terms of, if you get a blockage, what effect does that have, and how can we characterize that and use that in our whole brain model? So you can see under a normal condition, so the first thing we've done is looked at what happens if you change upstream conditions and reduce the partial pressure of oxygen. So on the top, in the middle, you've got the normal condition, so your inlet partial pressure of oxygen is at a healthy level. And you can see that the tissue is nicely well supplied with oxygen. And that drops hugely unsurprisingly, if you drop the PO2 at inlet. So here we dropped it from 40 in the middle to 20 on the right. And on the bottom, we just have a few slices through to illustrate the variability that you find. Because of course you get local regions of quite high oxygen because it's being dumped in the tissue and it takes a while for it to diffuse. and also regions of much lower hypoxia, regions of much lower partial pressure of oxygen. So, and in fact, what we do with these is we use a Green's function approach. This is a well-known approach in the field. It would take me a little while to go through the maths, so I won't, but ask me if you have any questions about it. But again, it's based upon exactly the same blood flow model. It's just that now we're looking at oxygenation and the tissue response to changes in here in arterial partial pressure of oxygen. And in a moment, we'll look at the response when you start to block. In other words, mimic the results of microthrombi entering your microcirculation, which is an important part of understanding the global picture. Because if we block large vessels, that's in some ways easy to see because we can see them on the imaging, and then we just put the blockage in there and everything downstream is affected. It's much more complicated if we're looking at smaller changes. So, and this is work done by another of my graduate students. And here what he's done is plot the oxygen extraction fraction and oxygen consumption rate. So oxygen extraction fractions in orange and the metabolic rate is in purple as a function of PO2. And so on the left-hand side, he's plotted both of those. And the variability is by simulating lots and lots of these different statistical cubes, because we have all the properties of the cube. So all we do is generate lots and lots of them and run lots and lots of simulations and characterize their behavior. And interesting, you can see that actually the results don't depend hugely on the particular geometry. It's actually pretty consistent. The variability is quite small, which is a very important result, because it means that the response of the tissue will be pretty invariant depending on the properties of the cube. On the right, we then look what happens to the hypoxic fraction in orange and the average tissue partial pressure in blue. And the hypoxic fraction is basically the fraction of the tissue below 10 millimeters of mercury partial pressure. And this is important because this is what we can measure experimentally in the animal experiments that Ed and Sissy are working on in Amsterdam. So this is one way we can start to link the results to the animal experiments and the animal data that we're getting, that's being acquired. So the hypoxic fraction is a very useful metric. And we can see, well, this is what happens if we drop the inlet partial pressure, and that would happen because you get a blockage upstream. So this is a sort of a secondary effect. But we can also look at what happens if we block at this scale. So the previous slide is essentially looking at blockages further up in the larger vessels. This is now saying, well, what happens if I block vessels at this tiny little length scale, at the capillary scale? And so what we've done is said, well, if we have a certain blockage fraction, say if we block 10% of the vessels, how does the perfusion drop? So on the left-hand side, you can see the perfusion is 100% of baseline on the top left. This is all relative to normal values. And then as we block more and more vessels up to 20%, at which point it stops really being a network at all. And actually, really, you don't really get very much perfusion at all. The model really pretty much falls over at this point. And we can see we get a pretty straight line for this in terms of the blockage fraction generating the changes in perfusion. And what's interesting is that the slope of this line isn't 1, as you might expect. It's actually, it's about 3, which means for every 1% of vessels that you block, you reduce your perfusion by about 3%. And this is in really good agreement with other studies that have been published in the literature. So that was very encouraging when we got this result out. And likewise, the hypoxic fraction. Now, this is not linear. So you can block up to about 10% of the vessels, and really there's almost no hypoxia. But once you get above 10%, it's pretty linear, and then it goes up pretty quickly. So by the time you get to 20% blockage fraction, there's about 60% hypoxia. So perhaps unsurprisingly, this is really, really non-linear. And so this is a very important result, showing that actually at this length scale, you can lose about 10% of your capillary vessels before you really notice anything at all. But after that, it really starts to accelerate very quickly. And this sort of links back to the thoughts I mentioned looking at dementia, and the idea that actually as your capillary bed gets compromised, it takes a little while really before you really notice anything. But then after that, the acceleration is really quite brisk. So it's an interesting result with implications potentially in dementia, as well as looking at ischemic stroke. So we can characterize this behavior. And then we can, again, as I've done every time, we move up from our small scale. So we now move up to the next length scale. So this is now our voxel scale simulations, and these are Wabi's descending and ascending vessels. And the red planes just illustrate that this is a continuum model with the capillary vessels. And we can look, now in particular, we can look at how the behavior changes at different depths. So it depends, does it matter where your clot occurs or where your microthrombi appear? And yes, it does. On the right-hand side, you can see how the permeability changes depending upon where you put the microthrombi. And you can see it's really noticeable. And there are some regions where it's really important. So you can see about the third row down, the mean value, which is shown in this red star, is much higher than it is for any of the others. So it really matters where these microthrombi appear. And having run lots and lots of these simulations, and this paper's just been published, so that's great. And we have a lot of results from this, and we can simulate it at all these different planes. And that means we can then sort of get this map on the left-hand side, looking at how the permeability changes. So the permeability is on the z-axis as a function of grey matter depth, and also the volume fraction. So by running thousands of simulations, we can characterize the response. And this is really our homogenization approach. We run lots and lots of simulations and we average them out. And then that tells us how we can expand this and implement this to the whole brain model. Because on the right, we can now, these are looking at the probabilities of ischemia developing. So on the left hand of those three plots, so this is our perfusion deficit for a typical right middle cerebral artery occlusion. In the middle is our probability based upon the depth, which comes from the diagram on the left. And then if we combine those two, we now know the probability of infarction on the right-hand side for this perfusion deficit. So we can now look at that as a probability map if we have microthrombi of what the outcome is. So this is really neat because we can now start to look at the effects. And of course, they vary depending on the perfusion deficit, but they also vary upon where you are in the brain tissue. And the key thing about the multiple scales approach is that because we have all the properties encoded in our coefficients in the model, we can now simulate this accurately. And every point in space, we know what the properties are, and therefore we know what's happening at a capillary level. If we wished, we could zoom back in and look at the response. So ischemia leads to hypoxia. Hypoxia leads to necrosis and apoptosis. And there's a lot of validation we can do, but I'm not going to talk about that today because Ed and Sissy have done some really fantastic work on this, and they're going to present on another occasion. I don't want to steal all their thunder, but there's a lot of work underway between us and them to do this validation and to get as much data, as much information as we can out from the data. And so what this all leads to is that we can then start to predict the response of the whole brain, firstly to the stroke, and then on the left with no treatment, and on the right with treatment. And this is a very simple simulation, which I've played some times before, but I use it just to illustrate how all those 2D maps then turn into these 3D simulations. And we can actually scale up and simulate both with treatment and without treatment and look at the intactness of the tissue. And we're working on a more detailed model of this at the moment. So we can see how that all fits together. So what I want to do as I wrap up is talk a little bit about some of the work that's ongoing to develop this further. What I'm going to do, talk a little bit about the effects of aging on model parameters. There have been some very nice studies out recently looking at how age affects parameters that we use in our model. Looking at the effects of hypertension, that's one of the biggest risk factors. So we want to look at how does hypertension affect brain behavior, both in the short term and also in the longer term effects that hypertension has on the brain. Then think a little bit more about the virtual population. I'm not going to talk about this very much because Alphonse will talk about this at a later date. but that's very much where this work is heading, using the Mr. Clean database. And picking up very briefly on the thing I talked about earlier, the idea that the system controlling cerebral blood flow is very active and dynamic. So the effects of aging. So there's some very nice data out in the literature. These are from animal models, but it gets us started looking at volume density and area volume ratios. And that means that we can start to calculate how these vary. So the top row is young, the middle row is middle-aged, and the bottom row is elderly. And you can see how these parameters change at different stages in the life cycle of mice. And what we're doing, and we just had a paper accepted on this, thanks to Wabi and a project student we supervised together, looking at the effects of aging on cerebral blood flow. And that's something that we'll be looking to incorporate in the model. So essentially age then becomes a parameter in the model that you can tune and look at the response, and particularly including it in the virtual population, which will be very important. And even just looking at regions in the brain. So that's sort of, the previous slide sort of assumed, well, everything's the same everywhere. But actually, if you look at the effects of aging and look at the change in cerebral blood flow and the percentage change per year, in different regions, you can see it's actually quite different. So some regions actually is a much more robust decline. It's much smaller than in other regions, which are more vulnerable. And indeed, it's different in grey matter and white matter. And this is something that we will be able to put into the model by saying, okay, we know how this changes with age, and therefore we can start to simulate how the brain responds differently in different regions with aging. And this is a paper that we've submitted looking at this. So age will be an important factor as we progress forward, looking at how that affects the parameters. Hypertension, particularly because of the remodeling of the vasculature. As you can see, this is from a review paper a few years ago, sort of highlighting just some of the things that change, particularly looking at how autoregulation changes, how blood-brain barrier permeability changes, and how the vessel wall is remodeled. And so it will behave differently under the context of hypertension, and trying to understand how this behaves both for long-term hypertension and short-term hypertension, and what the overall effects of this are, and how we include that in the model is going to be a really interesting topic to tackle in the future. Then all the results I've shown you have really been on one brain, simply because it took so long to get the mesh that we've used that a great deal. And Tamas has now done a great deal of work in the last 6 months, expanding this to multiple brains and looking at larger groups. And this enables us to start including things such as collateral flow score and looking at the interpatient variability. And as you will know, if you've read the literature at all, the vasculature is very variable from one person to another. And in fact, the standard anatomy really is only what you find in about half the population. So trying to understand how we can include this in the model is going to be a really interesting thing to move forward as we look at generating these virtual populations in the future. And then finally, I haven't talked at all really about control of cerebral blood flow, which is actually how I came into this topic in the first place, because I was interested in how the brain controlled flow. And we know that that control is impaired in stroke. We know that that changes dynamically after stroke. And in fact, we know that if autoregulation improves, that's a marker of improved outcomes after ischemic stroke. So that's a very important thing to put in. And I put this slide up just to highlight how complex this is, because there are all of these different things. So there's the neurogenic response, the myogenic response, the metabolic response, and the sympathetic pathway, all looking to modify the vascular bed so that it controls cerebral blood flow. And simulating this, I have a number of graduate students looking at this. So traditionally, we've always assumed that this is done by arterioles. So this is diameter as a function of pressure. This is again animal data because it's what's available. And we assume that the myogenic response is what sets the diameter. And then the metabolic response counterbalances that, and it's the trade-off between these two that give us this relatively flat diameter as a function of pressure, the balance between the myogenic and metabolic responses. And so there are a number of key substances involved, but I'm going to skip over that because there's a lot of interest now in parasites. And I talked about parasites a short while ago, but there's a lot of interest in looking at the role of parasites in autoregulation. And I have a graduate student who's just finishing her thesis on this. And actually, the role of parasites looks much bigger than we've always thought. But that means control is happening at a very local scale. And that actually it might be the local conditions that are driving the more global control of blood flow, because it seems that the capillaries respond immediately, and then it's messengers that they send back that actually then control arterioles and so on, and moving up the system. And there's a lot of work still to be done on this, understanding how parasites behave and how they respond. And then constructing that and putting that, I'm going to skip over these last few slides in interest of time, and then think a little bit about how we use these models of control and then couple them with other models. So on the right is a model of the neurovascular coupling response, and have a graduate student who's looking at how we couple that with the autoregulation response to try and understand better how blood flow is controlled under different conditions and in response to all those different stimuli I showed you right back at the beginning of the talk. So hopefully I persuaded you how we can develop models of flow, blood flow and oxygen transport in the cerebral vasculature and tissue. And this is all based upon rigorous mathematics and models that scale multiple scales, which means that these models, because they bridge multiple scales, they require multiple compartments. But I also want to highlight the fact that because of this, it means that we can do validation at multiple scales as well, because all our models are based upon one scale, characterized and scaled up. It means that we can do the validation at all of these different scales. And that's one of the critical advantages of multi-scale modeling, is that you can do the validation across all the different length scales. And the cross-coupling between these scales is key to how they behave, and also the structure and function relationship is vital. So understanding how the structure of the vasculature and its function relate to each other is very important. And one thing I've not talked about at all, but I still have in mind to do at some point, is looking at multiple time scales, because of course there are some processes that are occurring at seconds, some at minutes, and of course for a patient these things can be occurring very slowly at this time scale of hours. And can we use the same kind of separation of scales on a length basis to look at the response and separate them out for different time scales? And that's something that we'll be exploring in the future, I hope. So I'm just going to wrap up by saying this is just a small subset of the people who've been working on these projects. There's a lot more, and a lot of great graduate students. And particularly, I wanted to just praise the two postdocs on the project. Well, Wabi has now moved on, but all his contributions, and Tamas, who's done, and both of them have done amazing work on this project. And so much of what I've just shown you wouldn't be possible without their work. And if you do have any questions, I'll be happy to take them, but I'm also going to say that you can read these two books if you're interested in reading more widely on the topic. The left one on autoregulation and the right one more generally on cerebral blood flow metabolism and how we model it and how we measure it and how it's controlled. And I have reached the end, and I think we have a few minutes for questions. So I'm going to unshare my screen and ask if anybody's got any questions. And thank you all for listening.
Thanks a lot, Stephen. It was really impressive work.
Oh, thank you. Thank you.
I would like to ask especially the PhDs, of course, to ask questions.
Well, maybe I have a question myself to say. So very interesting and very impressive talk, Stefan. Thank you very much.
Thank you.
So maybe I'm a bit confused when you couple all the scale. So I guess you have boundary condition that you have to impose on all these models. I'm not sure I understand it, or you mentioned it.
I skipped over that very, very quickly, because you're right. It's how you set the boundary conditions at your local. You have to make assumptions about the boundary conditions at the local scale so that you can actually solve it at the global scale. And so what we do is we assume everything is periodic.
Okay.
Because this is what everybody does, and because nobody's thought of any better way of doing it yet. But yeah, it's one of those things that everybody knows. As soon as you open the lid, the periodic boundary conditions peer out at you, and everybody knows that that's an assumption. It's a bit of a limitation, but until someone comes along and does a better job.
Okay, but if it works, that's already good.
Well, it seems to work, yeah. And if it's the best thing available,
Yeah.
then we go with it, and we work with it, and I talk to mathematicians, and they say, well, we're thinking about it. So
Okay, and about the computer effort was not so obvious to me how difficult this is. I mean, you said it's always simple equation, but I guess you have a lot. You have many scales. So globally, can you comment on the resources you need for that?
yeah, I mean, the advantage of doing the scaling is that it's much quicker to solve. So you can solve these on a desktop actually relatively quickly. Whereas if you were trying to solve the whole flow field, it would just be impossible.
Yeah, I mean, sure.
So yes, I mean, we'd sort of deliberately try and do it so that you could actually run this. Any of my graduate students can run this on their desktop.
Okay.
So, which is
really within
within minutes. Yeah. So it's very neat. I mean.
Yeah, it's very impressive.
Yeah.
So I was wondering about the edema modeling. What is the mechanism that you would like to approach next? I think it can be developed a little bit more. And I'm pretty sure that the whole edema process is very complex. What would be the first thing that you would like to address?
Yeah, I mean, there's been a lot of work on aquaporins actually in the literature, and that's something I would like to get a graduate student to look at properly and see what data do we have, and can we scale that up properly, because we're still not really. The advantage with blood flow, which is why we started with it, is there's a lot of physics involved.
Yeah.
So there's a lot of equations it really has to obey, and in fact with water transport that's true, but there's a lot of biology going on that's not very well understood yet.
Yeah,
so, which is sort of why I've never really pushed on it a great deal, because the foundations are not as solid, I think, with that. So I would love to do it, and but it's the kind of thing I'd love to collaborate with an experimentalist and try and understand what they can tell us about these processes, because I don't know so much about it.
Yeah, it might also be a little bit become more important in the near future. We're not for sure. We don't know for sure, but yeah, it could be.
Yeah, no, exactly. I agree.
Any more questions?
Great. Well, I realize it's nearly 6 o'clock your time, so it's only 5 o'clock here in the UK. So.
Okay. Yeah. In Holland we have dinner at 6. So.